So, good afternoon. Welcome to our session to talk about how to improve drug tolerance of competitive ligand binding assays for NAB assays. We are here together, my colleague and I, but we are separated by a wall due to the corona rules. And even though we're tested, we have this as an additional precaution. So we're all safe. So let me introduce myself. I'm Regina Bruyns. I'm an immunologist with a long-standing experience in cellular immunology, flow cytometry, and ligand binding assays. I began my academic research in the field of tumor immunology at the German Cancer Research Center in Heidelberg, followed by a PhD on early lymphoid development at the Max Planck Institute for Biology in Tübingen. From there, I went for a postdoc to the Leineberger Cancer Research Center in North Carolina, working on hematopoietic differentiation of embryonic stem cells. On returning back to Germany, I joined the Department of Molecular Hematology in the University of Frankfurt. And this was then followed by working for CROs, initially for Focus. And after this company was acquired by Nuvisan, I joined Nuvisan in 2011. Here I built up the labs for cell-based assays and flow cytometry, as well as for immunogenicity assessments. And now I pass over to my colleague.
Good afternoon, I'm Sripriya Jaganathan. I work as a Bioanalytical Scientist in the Immunoassay department of Nuvisan Pharma Services. My current activities in Nuvisan focus on developing immunoassays to detect anti-drug-antibodies and neutralizing antibodies. Regarding my background, I have a Master and Master of Philosophy in Biotechnology. Prior to joining Nuvisan, I worked as a researcher in University of Ulm in multidisciplinary projects ranging from biology to nanomedicine.
So we will explain the house rules again. All your microphones are muted. If you have any questions, please use the Q&A chat box, and the recording and the slides will be sent to you after the talk. Let me introduce Nuvisan. Nuvisan is a full-service CRO, accompanying you from drug discovery to phase 4. We have a very low turnover in staff, and this assures that once a project manager has been assigned to your project, it will accompany you for a substantial period of time. So we will now have a poll asking you whether you have been working with Nuvisan before.
I think the people who are employed in Nuvisan may not have to answer this poll. Oh, yeah.
So we see the results coming in right now.
Yes.
Yes. And there are quite a few who have never worked with Nuvisan and who will try to convince today to change this.
Hopefully.
So, yeah, very good. Thank you for answering the question.
Okay,
so within Nuvisan, the department IMM, where IMM stands for Immunoassays and Immunology, has a head count of 32 people. And 11 of those people are holding a PhD, which is very helpful for research and development experiment studies. Our staff is highly qualified and we can accommodate a great variety of needs, starting from cell culture work, flow cytometry, immunogenicity, biomarkers, up to high throughput study sample analysis with up to 10,000 samples. Also, we have a lot of experience in biosimilar work. So as already mentioned, one of our services is the immunogenicity assessment. And these assessments are necessary if the medication is a biotherapeutic. Given that such biotherapeutics are present at high serum levels over a long period of time, it is not always possible to collect samples at trough levels. And this means that we have to find drug removal strategies And these are very important to be used in immunogenicity assessments. Today, we will show you how we managed to improve the drug tolerance for a competitive ligand binding assay that is used for the detection of neutralizing antibodies.
Okay, so let's start with a brief background about immunogenicity in a nutshell for all those who are not familiar with this field. When you administer therapeutics to humans, our body generates a wanted immune response. For instance, if you give a drug to kill cancer cell, our body generates immune response that is beneficial to kill the cancer cells. In addition to this wanted immune response, there is unwanted immunogenicity, in which the antigen presenting cells like dendritic cells will present these peptides as specialized peptides that activates the T cells, which in turn generates anti-drug antibodies. In short, we call them as ADA. Now coming to neutralizing antibodies. Neutralizing antibodies are a specific type of ADA that can interfere with drug-target interaction. These antibodies can have an impact on pharmacokinetics of drug, and they can also reduce the efficacy of drug by specifically targeting the domain of the drug that is responsible for its therapeutic effect. In addition to this, the antibodies are not only generated against the therapeutics, but also are generated against the endogenous counterpart of therapeutics, which may result in advanced clinical effects. So in order to know that the drug you give to the humans are safer, regulatory authorities have requested immunogenicity assessment, for which we use a three-tiered approach. The first approach is a screening assay, where you screen the samples for the presence of anti-drug antibodies. The positive samples are then confirmed by confirmatory assays, followed by NAB assays to know the presence of neutralizing antibodies. And now, regarding the detection of neutralizing antibodies, we have two formats. One is a cell-based format and another is a competitive ligand binding assay format. It is important to know your drug's mode of action in order to know which of these formats you can choose for your drug. If you'd like to know more about which of these formats are suitable for your drug, you can refer to Wu et al. But nevertheless, in this presentation, we are going to focus only on competitive ligand binding assays. There are two different formats in competitive ligand binding assay. The first one is a drug target code format, and the second one is a drug code format. In drug target code format, here we coat the surface of the plate with target, and we detect using sulfur-labeled drug. In the presence of neutralizing antibodies, we could see that the neutralizing antibodies will bind to sulfur-labeled drug, and there will be a signal decrease. In the case of drug code format, we coat the surface of the plate with drug, and here we detect using sulfur-labeled target. And in presence of neutralizing antibodies, they can bind to the surface of the drug coated on the surface of the plate, reducing the signal. There is also a possibility that you can biotin label your target and detect using streptavidin. In this case, you could see that the neutralizing antibodies will bind to the drug on the surface of the plate and thereby inhibit the biotin label target binding to the drug. Okay, so the main challenge in developing neutralizing antibody assays are drug interference. Why it is important to develop an assay that has an increased drug tolerance? It is because the sample or matrix which you receive have high amount of circulating drugs that are soluble and that can form complexes with NAB and prevent the detection of NAB in your assay. In this scenario, if you could see, this is a drug target code format, and if you have this residual drug present in your sample, the residual drug can bind to the target on the X-ray plate, and as a result, you have a signal decrease, which gives you a positive result, that is false positive result. In the same way, in the drug code format, the residual drug can bind to your sulfur label target, and you have less signal, which gives you false positive results. Okay, now let's see what happens if your sample has both neutralizing antibodies and residual drug. This is a drug target code format. Here you could see that the residual drug present in neutralizing antibodies can form complexes with drug, and as a result you have a signal increase which would give you a false negative result. So now it is very clear that if you need an assay with increased drug tolerance, it is important to remove the drug in your sample matrix. In order to remove the drug in your matrix, there are various strategies available. For instance, acid dissociation, affinity capture elution, solid phase extraction with acid dissociation, which in short we term it as SPEED, precipitation and acid dissociation, it's a PNA, and the last one is biotin drug extraction and acid dissociation, which we call it as BEAD.
So we have a second poll now, and we would like to know what what you see as the main challenges in the immunogenicity assessments and in neutralizing antibody assays, especially. So we have some guesses that we took before.
Yes. Let's see if you are right and if I am right.
Yes, and I think my guess is coming true. That is that most of you
Yes.
are interested in drug tolerance, and I think that's why you're here.
Yes.
So, yeah. What else? There are more obstacles to overcome here, yeah. So, this is looking interesting. And it evens out a bit.
Yeah.
Good.
We also have drug target interference.
Yeah, of course, but we're not going to touch on this today. We just stick to one topic. So the main topic is drug tolerance, as suggested. Yeah, fine.
So, yes, the results of the poll state that most of you have problems in improving the drug tolerance. So let's continue to see what we have to offer for you for drug tolerance. Okay, in this session, we would like to take you through our NAB assay development journey with drug X. And drug X is a monoclonal antibody, which is a biosimilar. Initial experiments with drug X include selection of suitable positive control and format. A format that is not prone to produce false positive or negative results was chosen in the presence of drug. And we have also evaluated different concentrations of coating and detection reagents in the presence of different concentration of drug. We have already tried all the different methods I stated before in order to see if we could improve the drug tolerance for drug X. And this table here shows the different methods we tried and the results we obtained. Here you could see that in acid dissociation, we used 300 millimolar acetic acid and 6% acetic acid, and we could not improve the results because we have less sensitivity, and we cannot improve the sensitivity and drug tolerance. We also followed a speed method using 300 millimolar acetic acid and excess of biotin labeled drug. Here, the sensitivity is not improved, and the equimolar concentrations of drug and PC, which was tolerated in acid dissociation, was not tolerated by speed experiment. Next, we tried the bead method using streptavidin coated beads, and here the sensitivity and drug tolerance is not improved. Next, we moved to drug depletion strategies by using positive control coupled to magnetic beads. Here the sensitivity was okay, but the drug tolerance is not improved. So as a next step,